Thank you for the introduction, Mansi. So, hello everyone. I'm Soumya, and today I'm going to talk about my work, my PhD work that I did like a few years ago. And currently, I'm not working in the Indian Ocean region, so I thought this would be more appropriate for the talk today. So today I'm going to be talking to you about the contrasting phytoplankton and biogeochemical functioning in the Eastern Arabian Sea shelf waters, which is also the west coast of India. And here we looked at how the geochemical functioning influences the carbon isotopic signature of the particulate organic carbon, which has not been looked at before in this region. So I begin with a brief introduction for the monsoons that is characteristic of this region. So I'm sure that most of us here are aware of the monsoon, so I'm not going to get into details and quickly move through this. So monsoons are this seasonal reversal of the winds that takes place in this region. And it happens twice a year, once during winters and then once during summers. So during winters, the intertropical convergence zone, where the trade winds from the north and the south, they meet, it lies closer to the equator. So if you look at the picture here, we see that I cannot see my pointer, but yeah, in this first picture here in winter, we see that the direction of the winds is mostly northeasterly. And then during the summers, when the the northern hemisphere, it heats up, the intertropical convergence zone, it moves to the north of the equator. So during this season, the winds are primarily southwesterly in direction, and this happens during the month of June to September. So these southwesterly winds, because they're blowing from the ocean to the land, they bring a lot of rainfall in the Indian subcontinent. So it's not only the Indian subcontinent, where it receives a lot of rainfall, but it's also like a special season for the Arabian Sea itself. And that is because during this season, we see it's the highest productivity that we see in the Arabian Sea during this season. So these southwesterly winds, they result in a very strong upwelling that is seen along the coast of Somalia and Oman. And there's also some upwelling that is observed in the northern part of this basin, which is the open ocean upwelling, but I'm not going to get into that. But coming to the eastern margin of this basin or the western coast of India, we see that the upwelling, which is moderate to weak in intensity, is observed in the southwest part of this, in this coast, but the upwelling signature is not present in the northern part of this shelf. So there's this contrast in the physical forcing between the south and the north in this region. So if we look at the wind pattern here in my third picture, we see that winds in the northern part are primarily, they're perpendicular to the coast. So they bring a lot of high salinity waters closer to the coast, which prevents these nutrient-rich upwelling waters from reaching the surface. Whereas if we look at the wind direction in the southern part, we see that the winds begin to turn, and the strong alongshore component of these winds, they takes the surface water away, which is then replaced by these nutrient-rich, waters from deeper depths. So that is one of the reasons why we see upwelling in this region, though there are also studies by physical oceanographers in this region that have shown that it's not only the alongshore winds that are responsible for upwelling, but there's also some remotely forced Kelvin and Rossby waves, but I'm not going to get into the physical oceanography here. So we wanted to see the influence of this contrasting physical, chemical, or the physical forcing on the chemical and the physical parameters here. So what we did was we sampled these 14 stations, which lie on the 200-meter isobath in the west coast of India, and they're about 1 degree apart. And this transect is very similar to what was done in the joint global ocean flux studies in 1994 and 1995. And we followed a similar transect just in case if we wanted to compare it to what was done like 2 decades back. So here, like I mentioned earlier, in the south, we see this upwelling, which is limited to about 12 degrees north. And then north of that, we do not see the upwelling signature. So from now on, these 5 southernmost stations, I'm going to refer to them as the southern stations and then rest of it as northern stations. So if you look at the physical, parameters or how they're vertically distributed. So in these kind of graphs here, I've put depth on the y-axis, and then I have latitude on the x-axis. And when I move from left towards right in the graph, I'm moving from south towards the north on the map. So I'm moving from, this along the west coast of India from south to north. So if you look at salinity, like I said, there's high salinity waters in the north because of this, the perpendicular winds that bring in a lot of high salinity waters. And then in the south, we see the salinity is lower. And then there's also this low salinity patch that forms because of the high rainfall and river discharge during this period. And if we look at temperature, the upwelling signature is even more clear. So we see these cold waters that move closer to the surface. But if you look in the north, we see that this thermocline is much deeper at about 40 meters or a little bit more deeper than that. Now, based on this, we looked at the nutrient distribution and the mixed layer depths. So the mixed layer depths are consequently shallower in the south, and then they are much deeper, like the thermocline at the northern stations. And here I've looked at the distribution of dissolved, nitrate and nitrite, and then dissolved inorganic phosphate. So we see that these nutrient-rich waters reach closer to the surface in the south, whereas the nutricline for both of these nutrients is quite deeper. Here I've not shown dissolved inorganic silicate, but even silicate has a similar pattern as to other inorganic nutrients. Now, as expected, or keeping in line with this distribution of physicochemical parameters, we looked at the chlorophyll and particulate organic carbon that we believe is mostly produced by the phytoplankton. So we see, or here in these graphs, I've taken the values, average values between the mixed layers and then what we see below the mixed layers. So for chlorophyll and for particulate organic carbon as well, we see that in the south, the values are higher. And then there's a sharp decline in the values as we move towards the north. And that is something we would expect because of the way the nutrients are distributed. But interestingly, we see that at the base of the mixed layers at the southern station, there's also a significant decline in both the chlorophyll and the particulate organic carbon values. We'll get into why that happens in some time. And along with the particulate organic carbon, the particulate nitrogen also shows a very similar pattern, which you can understand from looking at the correlation between these two parameters, which seems to be quite excellent, in the entire transect, which also points to the similar source for these two, elements. So before I get into how the isotopic signature of the particulate organic matter varies in this transect, I will briefly introduce isotopic fractionation. Again, I'm sure a lot of people already know about it, so I'm going to keep it brief. So isotopic fractionation, it mainly arises because of how the isotopes of element are utilized in a natural process. So, and this differentiation between how they're utilized results from the differences in their masses because of the difference in the number of neutrons and also in their thermodynamics and kinetic properties. So in a reaction, there would be a certain ratio of okay, for example, I take the example of carbon here. So carbon has two stable isotopes. There's carbon 12 and there's carbon 13, 13 being the heavier isotope. And in most of the reactions, we see that there is a preferential utilization of the lighter isotope. There can be, it can happen otherwise also. There are some reactions that, researchers have seen that where a preferential uptake of heavier isotope might take place. But for the purposes of, for the context of my presentation, I would say that there's a preferential utilization of a lighter isotope, which leads to the difference in the abundance of these isotopes in the substrate and the product. And now because there's a utilization or preferential utilization of lighter isotope, then the ratios of the heavier to lighter isotope tends to be lower in the product when I compare it to the substrate. So this difference in the abundance of the ratio is what we call isotopic fractionation. And one very, great example of where this takes place is photosynthesis, wherein the inorganic carbon, which can be carbon dioxide or bicarbonate or carbonate ions that are taken up by these phytoplankton cells or even plant cells, and then they're converted into organic carbon. So this photosynthetic pathway, it leads to a substantial fractionation between the substrate, which is the inorganic carbon, and the product, which is the organic carbon. Now, an interesting thing here is that the different kind of photosynthetic pathways that are used by biological organism, they result in distinct ratios between these two isotopes. And so yeah, it would become a little bit more clearer if you look at the picture here. So we have C3 plants. So C3 plants, they mainly utilize rubisco, and rubisco leads to a lot of isotopic fractionation. That's why for C3 plants, we see such lower values, By lower, I mean more negative values. So for C3 plants, we see values close to -30 to -35. Then for C4 plants, which use PEP carboxylase as a primary, carboxylating agent. So for them, the fractionation rate is lower, and that's why we see less negative or higher values as compared to the C3 plants. So the values would be somewhere between -15 to -10. And then there is marine algae, which lies somewhere in between the C3 and C4 plants, and then it has a different photosynthetic pathway. It could also have mechanisms like carbon concentrating mechanisms that result in such a signature. So it seems that there is quite a different, isotopic signature for these different biological organisms. And this becomes a very useful tool when we're trying to ascertain or delineate the sources of organic matter, particularly in a coastal region, because in the coastal region, the organic matter could be coming from various different sources. There could be phytoplankton, producing organic matter in situ, and then there could be this terrestrial organic matter that comes through different rivers and creeks. So this is quite a powerful tool for delineating the sources of organic material. Now I come back to my results. So if we look at the distribution of delta 13C of the particulate organic carbon in this region, we see a similar pattern that we saw for chlorophyll and particulate organic carbon. So if you look at the southern stations here, we see the values are higher or less negative compared to the values elsewhere. And there's a clear contrast between the south and the north, where in the north we see lower values. And even at the base of the mixed layers of the southern stations, we see significantly lower values. Now, if we were to go by what is traditionally assigned to the different biological organisms, we would say that, okay, these values in the lower values lie between -29 to -26, which is what we usually observe for C3 terrestrial plants. And then we say, okay, at the southern stations, we have values close to -23 to 20, which is what we observe for marine phytoplankton, which kind of makes sense for the southern stations because there's a lot of nutrients. So microphytoplankton, particularly diatoms, would take advantage of that and would be present in abundance. But at the northern station, it kind of doesn't make sense for our study area to have a lot of organic, terrestrial organic material because the western shelf of India is quite broad, particularly in the northern part, where it can be as broad as 180 kilometers. So, and there's also no major rivers that bring in a lot of terrestrial material. And then there is this supporting evidence. So if you look at C/N ratios, we see that most of our ratios, particularly in the south, they're close to the canonical Redfield ratios. But even in the north, they don't really exceed 13 or 12. But if we had terrestrial organic material that is more rich in lignin, which has a lot of carbon, the C/N ratios for terrestrial material are usually much higher than 20. So the C/N ratios has a different story. And then if we look at particulate organic carbon and chlorophyll concentration, there seems to be a significant positive correlation between these two parameters, which again tells us that POC seems to be produced in the same way that chlorophyll is being produced by marine phytoplankton. So there seems to be different reason other than the terrestrial organic matter for such low values that we see in our system. So we start looking at what these different factors could be. So first we explored the phytoplankton community composition. So here I have these different size classes, with the microphytoplankton being the largest, picophytoplankton being the smallest, and nanophytoplankton somewhere in between. These have been constructed using pigment analysis that was done in HPLC. So if you look at the community composition at the surface, which is my top panel here. And here I'm taking the surface as a representation for what is happening within the mixed layers, because we saw that the phytoplankton community does not change a lot within the mixed layers. So if you look at the southern stations, we see there's a greater contribution of microphytoplankton to the community, whereas in the north, we see there's a greater contribution of smaller phytoplankton, such as picophytoplankton, to the phytoplankton community. So there's a contrast in the phytoplankton communities between the north and the southern station, which makes sense when we look at the nutrient distribution, because the larger diatoms or larger phytoplankton such as diatoms, they tend to perform better in nutrient-rich waters, whereas in oligotrophic waters, smaller cells, because of their larger surface areas to volume ratios, they tend to perform better. So the phytoplankton community composition kind of makes sense to us. And it has been seen in a lot of lab studies that there is a significant difference in the isotopic signature of these large and small phytoplankton. So the larger phytoplankton, particularly diatoms, they tend to have this IA form of Rubisco, which fractionates less and hence would lead to higher delta 13C POC values, whereas the smaller phytoplankton, they have a ID form of Rubisco, which tends to fractionate more and could be one of the reasons why we see lower delta 13C POC values at the northern stations. Now coming to about 40 meters, which I take as a representation for what is happening below the mixed layers. So we see there's a significant decline in the microphytoplankton in the northern stations, whereas there's a simultaneous increase in pico and nanophytoplankton abundance at 40 meters or below the mixed layers at the southern station. At the northern station, there doesn't seem to be a significant change with the depth in the phytoplankton community composition. So with the depth also, there seems to be increase in these smaller phytoplankton types that have higher fractionation, which would lead to the lower values that we see at the base of the mixed layers. Next, we try to look into the community a little bit better using the pigment correlations. So if we look at the southern stations, I have fucoxanthin here, which is a diagnostic pigment for dye atoms. So we see the fucoxanthin shows a significant positive correlation with chlorophyll. And this tells us that dye atoms are the major contributor to chlorophyll in the southern station. And this has also been confirmed by a lot of microscopic studies. In the northern stations too, we see that fucoxanthin shows a significant relation with chlorophyll, but there's a difference in the slope between what we see in the south and the north. So in the northern stations, it seemed there could be a dominance of smaller diatoms because of the lower nutrient concentrations that we see there. Next, if we look at 19HF, which is a marker pigment for these nanophytoplankton, which is known as coccolithophores. So we see at the southern stations, there's a poor correlation between chlorophyll and this 19HF, which tells us that coccolithophores may not be that dominant actually in the southern stations, but in the north, we see this better correlation between these two pigments, which tells us that these smaller cells are dominating at the oligotrophic northern stations. So there's clearly a change in the phytoplankton community between the two regions. Next, we come to the growth rate. So for growth rate, I've used this model that was developed by Lee et al. For the Californian waters. And here the growth rate is calculated using a function for nutrients where there's a co-limitation between nitrogen and silicate. But for our region, particularly in the northern region, we mostly see a limitation of nitrogen. Then there's a function for temperature and then another function for light or photosynthetically available radiation. So using the values that we see in our data set, we see that in south, there's a significantly higher growth rate. And then in the north, the growth rate declines significantly. And interestingly, we also see that the growth rates for the base of the mixed layers is lower than what we see in the mixed layers at the south. But in the north, it's quite interesting to see that the growth rates at the base of the mixed layers are slightly higher than what we observed within the mixed layers, which could happen because of the temperatures. So the temperatures are quite high, which are known to slow down the growth rates because the respiration also increases with higher temperatures. So that could be one of the reasons why we see such a difference in the growth rates. So, but the main point here is that even the growth rates have, in the laboratory, it's been shown that even the difference in the growth rate leads to a difference in the isotopic signature of the particulate organic matter. The slower growth rates, they actually increase the rate of fractionation, whereas when you have faster growth rates, particularly in organisms like diatoms, which have higher carbon demands, the isotopic fractionation tends to be less. So growth rate seems to be also one of the reasons why we see such a difference. And at the base of the mixed layers also, there's a decline in this growth rates where we also see a decline in the delta 13C POC values. Now coming to another factor, which is light limitation. So for light limitation, what we did was we used this ratio between the photoprotective pigment and light harvesting pigments. And then we tried to look at how this varies between the south and the north. So in the south, we see the ratios between these pigments was low. So a lower ratio would mean either the light harvesting pigments are higher or there is a lower production of photoprotective pigment. So they're producing more light harvesting pigment and then there's no photoprotective pigment, which means that there could be a light limitation in the south, which is something we would expect because the chlorophyll concentrations are so high that would attenuate light much faster in south as compared to the north. So if we look at the northern station, these ratios are higher, which tells us that either the light harvesting pigment is being produced, there's a lower production of light harvesting pigment, or there's a greater production of photoprotective pigment that a cell would do to, protect itself from photo damage. So in the south, there seems to be a light limitation, or at least the growth is somehow controlled by this light availability. But in the north, where the waters will be more clearer because they're not as productive, light does not seem to be limiting, particularly within the mixed layers. That may change at the base of the mixed layers because that becomes quite deep. So if we look at the ratio between these pigments and then the delta 13C POC, we see in the south, there seems to be a significant correlation between these two parameters. But in the north, because there doesn't seem to be the influence of the light in controlling the growth of the phytoplankton, we don't see a correlation between these two. So light may not be directly, influencing the delta 13C POC values, but it could indirectly influence these values by, controlling the growth rates or other metabolic functions of the phytoplankton. Again, now I come to my last parameter, which is the heterotrophic degradation. So it's difficult to observe heterotrophic degradation in the field and also during sampling. So I just looked at the kind of data we have, and then we tried to perform some experiments to see how degradation influences the delta 13C POC values. So first of all, if we look at here we try to calculate the photic depth by using the total pigment that was available. So we look at how the values vary within the photic depth, and then we see how it varies below it. So for most of these stations along the west coast of India, we see that delta 13C POC values are actually slightly higher within the euphotic depth, and then they decrease when we move below the euphotic depth. We did a similar exercise with the similar transect when we sampled it again in August 2018, and we see that even though the change is not as drastic as what we observed in 17, but for most of these stations, there seems to be lower values below the euphotic depth. Now, this is also something that has been observed recently by Close and Henderson, and they put it in a paper in 2020. So even though this was observed in open oceans, they see that there is a significant decline as we move down the water column in delta 13C POC values. Like if you see here in these graphs, there's a decline of about 2 to 4 per mil. This decline happens at much deeper, depth in the open oceans than what we observed in our coastal samples. So even though we don't really understand what's happening, so we try to sort of, perform these lab experiments to see if degradation is actually influencing these values. So we did this simple experiment. Here I just have a very simplified scheme of the experiment. So we collected some water, seawater, and then we kept a part of it to preserve the original phytoplankton community, and then we filtered the most of it, and then in the filtered seawater we put in some nutrients and then added the phytoplankton community to let it grow so that we know how much organic material we have to begin with. And then after letting it sit in the natural daylight cycle for about 3 hours, we had a good phytoplankton bloom, and then we kept those bottles in the dark to let them degrade, and then it sampled it over certain time intervals. So we did 2 such experiments. So the first experiment was conducted over 6 days, and then the second one was conducted a little bit longer for 18 days. So we see that if you look at the particulate organic carbon, it decreases as with the time, which is something we would expect because of degradation. But interestingly, we see that with the degradation in POC, there's a significant decline in delta 13C POC values during both our experiments. And even more interesting is that in the second experiment, we see that the degradation rate, it seems to decline after the 6th or after first week, probably because of this accumulation of refractory material, which is difficult to break down. So there seems to be a decline in the growth rate, and this decline in the growth rate is accompanied by this constant or more or less constant values of delta 13C POC values. So even though we don't understand what exactly is happening, why are these values declining, but we know that degradation has a role to play. Now, this is something that has also been observed in sediments, actually. So in the sediments, it's been seen that as the percentage of total organic carbon decreases, the delta 13C POC values also start declining. So in some of the literature that I was looking into, the researchers or the scientists say that that probably is happening because components of organic materials such as carbohydrates and amino acids, they tend to break down much faster, and these components tend to have higher delta 13C POC values. And what gets left behind is lipids, and lipids have lower delta 13C POC values. So that's why as the carbohydrates and amino acids, they're consumed, lipids, get left behind, and they lower the delta 13C POC values of whatever is left behind. But this is something that still remains to be seen in the lab experiments, and probably it's also something that we could look into in future. So yeah, but there's clearly an influence of degradation that we see here. Now, with this, I sort of come to the conclusion, and I would like to summarize my presentation by saying that we see that in the southern stations, there's upwelling, there's a lot of nutrient that comes up, and this nutrient, that it promotes the growth of microphytoplankton that are also growing faster, which leads to this lower delta 13C POC values. Whereas in the north, there is oligotrophy, and which supports a slow growing smaller phytoplankton like picophytoplankton, nanophytoplankton, which lower the delta 13C POC values. And at the base of the mixed layers, even though we did not directly observe it, there seems to be an influence of degradation. So that is what we found. But with this, I would also like to say that there are some other factors that can lead to the differences in the values that we see. So first of all, is the isotopic signature of the source inorganic carbon, which is the CO2 or even the bicarbonates or carbonates. So we also need to see what is the isotopic ratios of the source inorganic carbon, which unfortunately we were not able to do. So that is one factor that could, influence the values also. But I was looking into some of the studies that have been done here, and I could find one such study, which is not done along the same transect. It is a little bit more offshore. But in those studies, the delta 13C of DIC is actually not so different. The difference is about 1 to 2 per mil only, but the difference that we see between north to south is more than 2 per mil. So even though there might be influence of the dissolved inorganic carbon, but clearly there are these other factors that have to be, considered. Then secondly, we also did not measure the partial pressure of CO2, which is also seen to influence the delta 13C POC values. But we did try to, relate these values with the temperature, because temperature controls the solubility of CO2 in the waters. So it doesn't seem that temperature really affects the delta 13C POC values. So it seems that the CO2 concentration might also not be a major factor in this region that influences the delta 13C POC values, even though I cannot completely rule that out. So with this, we had an important learning that what the values that are traditionally assigned to different groups of organisms may actually not apply everywhere, because these values are first of all mostly obtained from the temperate regions, and to utilize them to delineate the sources of organic matter, we actually must exactly know how the local sources are behaving. So if we would just assign these low values that we see in the north to terrestrial organic matter, that would not be correct. So yeah, so before utilizing this as a tool for delineating the sources of organic material, we exactly need to know how the local organic material varies before we can do that. Otherwise, we would actually be making a lot of mistakes with this. So yeah, with this, I end my presentation, and I would like to thank all of you for sparing some time and coming and listening to this presentation. And I would also like to acknowledge my PhD supervisor, Dr. Hemanti Biswas, and also my other lab mates and the technical assistants and the ship staff that helped with collecting these, the samples. And also without them, it would be really impossible to come up with a data set like this. So again, thank you, everybody. And yeah, with this, I end my presentation. Thank you.
Thanks, Soumya, for this amazing presentation. It was really good. So now we can take questions, and please feel free to unmute yourself and ask, or you can post it in the chat box. Yes, Matthew?
Thank you. Yeah, thank you for the great talk. I'm more of a physical oceanographer myself, so a lot of the biology goes past me. But I was wondering, is it possible to see these differences in the community structure with using remote sensing? Would that be possible?
Okay, I'm really not sure how that would work for diatoms, but I know that for coccolithophores, it is actually possible because they have this milky this color, milky turquoise color that is actually detectable from these satellites. So especially in like I'm working in the North Sea now, so I see a lot of my colleagues because there's a large coccolith bloom that forms in the North Sea, and it's actually detectable by the satellites. So I know it works for coccoliths, but yeah, I'm not sure if that actually works for diatoms. Yeah, I mean, you could definitely see the chlorophyll, but yeah, I don't know if you can use that to really delineate the composition. But yeah, for coccolith, it is possible.
Okay.
Yeah.
Thank you.
Any more questions?
Soumya, can you hear me?
Yes, I can hear you.
Very informative talk. Thank you for that. I have only one question. I have studied the Eastern Arabian Sea, right? So Eastern Arabian Sea, like generally, if I see, for example, if you go through your summary slide,
Yeah.
the southern station, you have mentioned like up to around 12 or 13 degree is mostly upwelling. You are attributed to upwelling, but if you look at the southern Eastern Arabian Sea, the upwelling goes up to like even the northernmost 19 degree, 20 degrees also there's an active upwelling, but it is a short term. But like Goa and like Ratnagiri and means up to 17 degree north, you can see very like intense upwelling, particularly during the summer monsoon season. So how you could like argue your, these results? Like for example, you have considered southern station as well as northern station. Like you have given only two divisions.
Yeah. So like I would also ask you like the upwelling that is observed in the northern region, because we're quite far away from the coast. That is also a thing. So maybe the one that you're referring to, the active upwelling that you see must be, I think, much closer to the coast. Is it so?
Northern station, it is much closer to coast. I do
Yeah.
agree.
Yeah.
But you see, like 13 degree to 15 or like a 16, 17 degree, it is like you can see far.
Yeah, and also I think it's also a little bit the way the physicists define upwelling and the way the biologists define upwelling. So for physicists, I think it's the tendency of the water to move up, which is also there actually here in our northern stations. But the way the biologists define it is when you see the productivity. So we don't really see that productivity at this distance from the coast. So I think that is also one difference. And also, yeah, again, what you're referring to is also, I think, much closer to the coast. So I'm only referring to what I see along this 200-meter isobath, where we don't really.
But as per like our observation, we got up to like we have collected monthly observations from up to 2000 meter contours. So we got like around 16 degree, 17 degree up to 16, 17 degree. We got like even 2000 meter. You can see the uplift sloping of isotopes during the summer.
And do you also see high chlorophyll there or something like that with these?
Chlorophyll, like I hope like there are much like some of the team members are in this presentation, so they have that.
Okay.
But I can say like I observed like around 15, 16 degree. Like I could say like the ocean, the low oxygen waters reach up to 40, 50 meter. Means like it is that much intense in the open ocean.
Yeah, so for here also we see that the thermocline is around 40-50 meters, but yeah, above that we see that there's quite low nutrients, and that's why we say that it's not non-upwelling. But yeah, I understand that the way physicists define it is also quite different from how, because for biologists it's, if you see high nutrients, there's high chlorophyll, and then we call it upwelling. So yeah.
Okay. So how about the virus in during the winter convection? No,
Can you please repeat that?
no, the effect of convecting thing.
Yeah, so in winters, that's what we see. Like this seems to reverse in winter, where in winters we see that in the south there's almost very low production, but in the northernmost stations around 18, 19, 20, 21 degrees, we see there is this winter convection, which brings in a lot of nutrients, and then the productivity increases. Yeah.
Yep. Okay. Thank you.
Thank you.
Others have no. Okay. Now, thank you for your nice presentation.
Thank you.
So now we have a question in the chat box by Biogeochemical. He's asking that can we differentiate the origin of POC specifically? Is it from phytoplankton or zooplankton?
Okay, so yeah, it becomes a little bit difficult to sort of differentiate between phytoplankton and zooplankton using just the delta 13C POC values because because there's not a lot of difference in, so it is that saying which we say that we are what we eat. So your isotopic ratio is going to be what you eat. So zooplankton mostly have the isotopic ratio very close to the phytoplankton. So that's a difference, but it's been seen that there's an enrichment of carbon that takes place. So zooplankton tend to have 1 or 2 per mil higher than the phytoplankton. So it is possible, but you would need C/N ratios also. And then it would be better if you have pigment ratio. So just, just using delta 13C POC for phytoplankton and zooplankton might not be very wise to do, but you would need other supporting data with it.
Any more questions from the audience who have joined us? I suppose there are more questions. Okay.
Am I audible?
Yes.
Yes.
Yes, I mean, this is Arvind. So it was a nice presentation. Actually, regarding this upbilling things, but you mentioned that in this southern station, upbilling is taking place, means playing a major role. So when we are talking about the upbilling of water, so there are different type of water masses which can come and upbill, and their properties are different. So which type of water mass you are talking about? Like suppose the deeper water is coming, or like intermediate or bottom, or some different kind of water masses, or the waters are getting upbuilt, because they all have their different phytochemical properties.
Yeah. So yeah, I'm not really sure, like, because I've not looked into water masses, if it's like bottom waters or immediate waters. But what I understood from the literature is that these waters that come up, they're actually not too deep. They come from about like 70 or 80 meters. So they're not really deep waters. But what we're concerned with here is that they have higher nutrients, which is good for productivity. And yeah, so that's what I know about the water masses. But yeah, it seems that they're actually not too deep. They're coming from 70 or 80 meters. So it's possibly just the regenerated nutrients that they have. And yeah, that is what matters for our study here. I'm really sorry, I don't have more.
It's fine, it's fine. But in that like top 100 meter or 150 meter, this dust can also play a major role, and we found that in the Eastern Arabian Sea or like in the open ocean. So then how will you differentiate whether this nutrient is coming from the building of the waters, and whether it is coming from the dust input or from the advection of the coastal, nutrient from the coastal sediment? Will you be able to find it? Yeah.
I'm sure that we can find that out, even though we've not looked at it. But and also, like, yes, in Arabian Sea, aerosols are an important source of nutrients. But if you look at I'm just thinking out loud. I'm saying that if you look at the wind direction during this time, it's mostly from the sea to the land. So I would say that aerosols probably are not a not a major contributor for this upwelling. So it's probably the regenerated nutrients from the water, because if you look at the wind direction, when you have aerosols or when you have these nutrients coming from the land, it's usually the winds blowing from the land towards the sea that would increase the productivity. But because the wind direction, I would say, I think that these aerosols might not be an important contributor during the southwest monsoon.
Okay, fine.
Yeah.
Thank you. Thanks so much.
Thank you.
Hi, Soumya, this is Medhavi.
Hi, Medhavi.
It was a very nice talk. I just wanted to ask one question, if you can elaborate that. You mentioned about the differences in delta 13C in sediments.
Yeah.
So I wanted to ask, like, how it will change the same thing, what you explained, how it will change in different phytoplankton communities if we have fluxes from different phytoplankton in sediment. And also, will it be only a matter if it's in the surface, or like we can go down core profile?
So I think this has been actually observed in core profiles only. Like you take a core and then measure the TOC percentage, I think, the total organic carbon percentage. And so in these profiles, I can send you those papers. So in these profiles, they've seen that as the TOC decreases, irrespective of the phytoplankton community. So irrespective of that, as the TOC starts decreasing, the delta 13C TOC also starts decreasing. So yeah, so I'm not sure if it really depends on the where the source or the community of the phytoplankton, but yeah, with the degradation there seems to be an influence irrespective of the community.
Thank you.
Okay.
Are there any more questions? I think there are no more questions. We want to thank you, Soumya, for this great presentation and then the great Q&A session as well. And this talk was recorded and it will be available on our YouTube channel. So everyone in the audience, or you can just refer to it if you want later. And yes, so with this, we conclude our today's seminar, Ocean Insight seminar, and we'll see you next month, first Friday of next month again. And thank you all for joining us. This is it. We can.
Thank you.
Thank you so much, everyone.